Viruses birth bacteria. It's like the worst version of Alien vs Predator. Foreign pathogens. Okay, first off, there's lots of differences between bacteria and viruses. And we're gonna just start off with this very specific stuff about bacteria. But you could probably predict a bunch of things that happen prior to getting going on this lecture. Also, shout out to my girl Kim, who also watches these, turns out. We have bacterial types, Archaebacteria and Eubacteria. Archaebacteria is a little bit more fun to learn about, like when we get into evolution. It's like archaea is literally named and coined after these being the original ancestors. And so it's like archaic times, literally. I want you to think of like early Earth. It's hot. There's like very high acidity. We literally called it a primordial soup. And so these type of bacteria, because they're direct descendants from bacteria from that early Earth, they're going to live in really extreme environments, extreme heat, extreme pH, and just good at dealing with harsh environments. That can be hot sulfuric springs, Great Lakes with high salinity, volcanic vents in the deep, deep oceans. Another thing is these are probably going to have RNA gene sequences instead of DNA gene sequences. Not necessarily true, but the sequences are definitely going to be different than what we find in eubacteria. Most of these are going to be autotrophs. They make their own energy, and it's going to be an anaerobic, but because there actually was not oxygen in early Earth, it ended up being made, and they're going to do this process called chemosynthesis, similar to photosynthesis, but deriving from chemicals as opposed to the sun. And last but not least, there's no peptidoglycan in the cell wall. Peptidoglycan is just a fancy name for the type of sugar, structural sugar. It is definitely going to be in its little beta formation of a carbohydrate, and it's a cell wall that makes up bacteria. Now, eubacteria is where we are going to find that peptidoglycan. It is the most familiar bacteria. These are decomposers, our mutualistic relationships, the gut bacteria that you guys know about, all of those ones. Those are good examples. There's also some bad disease causing ones, and usually they're the ones that give all that good bacteria a bad rap. They're classified via their gram stain results, positive or negative, and the shape of the bacteria and how it grows. We'll get to that on the next slide. These are ones that can obtain nutrients as either heterotrophs or autotrophs. It can literally be either one, meaning they can get their own energy or make their own energy as an autotroph, or they can have to obtain it. And then these guys, a lot of the times, have flagella. So here's how you name it. You're going to first say the grouping style, and then you're going to say the name. So this looks like a chain. So this would be Streptococcus because these are circular bacteria. This also looks like a chain to me. So it would be Streptobacillus. And then this one we're going to pretend like is a cluster or a colony because I haven't seen it yet. And I want to use the word. This would be a Staphylosporidium. And that is how you name things based off of their group and their shape. All right. Now, interesting things about eubacteria. The ones that are bad, we are looking for that peptidoglycan to be on the outside layer. This is a gram positive. So you got to think like this. If it is purple, it is positive for peptidoglycan on the outside, These are the ones that are easy to treat because they're not hiding from us. There are some that are red or pink. So these guys right here are super evolved bacteria. They actually have an extra lipid layer on the outside of that peptidoglycan cell wall, which will hide from antibiotics. The whole point of an antibiotic, it is going to bind to a bacteria, rip apart that peptidoglycan, and then it will be able to destroy it that way. Viruses, on the other hand, are very, very small. Please observe, here is a eukaryotic cell. This is the nucleus of the eukaryotic cell. This is a bacteria. This is a virus, even smaller than that. Now, the thing about viruses is they are not living things, although they do exhibit some qualities of life. They can evolve. They have a genome. They reproduce by hijacking the mechanism of the host cell that they are injected into. So it's a little bit different than a living thing because they don't have their own metabolic machinery. They don't have a way to obtain energy. They don't have a way to use energy. So they have to hijack a cell to do its bidding for it. Now, what they do is they hijack the pieces of the cell involved in DNA replication and protein synthesis. So that means those ribosomes, that means those enzymes like the polymerases that are replicating DNA. All of those are going to be used to reproduce a viral genome and make more proteins for the virus. And so these viruses are not considered living things. So main parts, it's got a capsid, fancy name for a protein coat on the outside, and then it's got a nucleic acid on the inside. It can be double-stranded DNA. It can be double-stranded DNA. It can be single-stranded DNA. It can be double-stranded RNA. It can be single-stranded RNA. If you see this one anywhere, you know it's a virus because that doesn't naturally occur in nature. If you see a single-stranded DNA, that's probably a virus because it also doesn't. We're going to talk about retroviruses in just a second, but they are very, very bad because they also come with their own cool enzyme, which does reverse transcriptase. How we name them is typically what is ever on the outside of them. So we got our coronavirus here, named for its crowns on the outside. Here's our proteins on the outside, and here is our genome on the inside. So two types of viruses, they're lytic and they're lysogenic. A coronavirus is actually a lytic virus. And so the word is virulent. They can be virulent, meaning they can be pretty much deadly. But they can also be non-virulent, where they're not actively like creating deadly versions of the virus. But this is a quick virus. It's gonna come, it's gonna hit you, and you typically have the ability to get over these types of viruses. They can be cold viruses, such as the flu. Corona is one example of this. They spread really, really quickly. They're actively reproducing and making more viruses. Versus the lysogenic virus. A lysogenic cycle is the DNA of the virus is actually embedded into the host cell's DNA. So this becomes a lifelong infection, a lifelong virus. It's going to be things like HIV or herpes. Chickenpox is a lysogenic virus. And the cool thing about these viruses is they go in and out of dormant states where they're lysogenic and they're hiding versus they enter a lytic cycle and they are actively reproducing more dangerous viruses. As viruses get their information transcribed and translated, what it's really doing is it's making more viruses, making more protein coats, making copies of the DNA to make those more viruses, but it's also making the proteins from the genome of the virus that are bad, like causing symptoms. So those two things are happening when you're in the lytic cycle. In the lysogenic cycle, it's just hiding the genome, not necessarily transcribing it. Now, an environmental factor can trigger a lysogenic virus to shoot into a lytic cycle. So here's a picture of the two. Here is just the lytic cycle, okay? Inserts DNA. DNA stays outside of the host genome. It's actively making more virus parts and lysing that cell and creating more viruses. They're going to go do the same thing. We also have the lysogenic one here. There's no active symptoms. It's in its dormant state, but the cells are reproducing. So there's lots of these cells all with the viral genome in it. Eventually, it will create into the lytic stage where we'll start making the viruses and releasing them. So treatment, and this is actually a good slide. We got, if it is a lytic virus, you're going to try to prevent it with a vaccine. This is a weakened or dead version of the virus, so our body can get immune to it or start to recognize it. This is going to come back up when we do immunity. and the immune response when we do systems. There's also antibodies, which is the actual immune system building up that recognition and being able to recognize and fight it. And then there's any antiseptics where we can destroy the outside surfaces. That's why they told you when there was corona going around to use hot water, because it would denature the outside protein and actually be able to destroy the coronavirus. Lysogenic viruses, all you can really do is treat the symptoms. And so what you want to do is try and reduce the cell processes involved with replicating that viral genome. So DNA replication is going to slow down, protein synthesis is going to slow down. Your antibodies will help you fight it off. Your antiseptic. Bacteria like bacteria that has peptidoglycan in it, that's the only treatable bacteria. And so target bacteria with antibiotics, and those antibiotics will rip apart the peptidoglycan. This is again only for gram-positive bacteria because the peptidoglycan's on the outside. For the gram-negative, our body's going to have to find it. That's our immune system, our antibodies, and antiseptics. Last but not least, we have bacteria that does not have peptidoglycan. You're probably not going to have to treat it because you're never going to come in contact with it because it lives somewhere you can't go. The last little thing about viruses and viral genomes is depending on the type of virus, we can call it a DNA or an RNA virus. And again, they're not cells, they're not living things. There's a wide variety of them, as you can see. And one of the biggest things is because of how good they are at hijacking, sometimes these viruses can combine with the host, not combine with a host, and then allow for mutations to kind of be more likely to occur. For instance, lots of clicking. For instance, when viruses are going through all of this cycle, they can actually express and alter the host phenotype. So what's going to happen is, here's what I'm looking for. If you're an RNA virus, there's not like a check system in place to check the DNA. Our body's actually really good at not messing up our DNA. We have 1, this is good, let's replicate, let's move on. 2, this is not good, let's fix it. And 3, this is not good, we cannot fix this, apoptosis. And so RNA viruses are actually really bad because there's no check systems on our RNA. So our RNA is constantly mutating. So if it's an RNA virus, it's more likely to mutate and evolve, and it's harder to get treatments for those because they're constantly evolving. And a retrovirus, what happens is it not only sends the viral genome as an RNA, but it also sends this little enzyme here, a reverse transcriptase, which actually changes that RNA back to DNA and then puts it in the genome. So, like I said, constantly changing, constantly evolving, constantly becoming something we can't recognize. Because of this, we can get this emergence of different viruses. This is how we got SARS. This is how we got corona. This is how we get mad cow disease. This is how we get smallpox, chickenpox, all the things. One of the things that is kind of unique is that this usually occurs when a new virus has the ability to expand its host territory. Meaning originally it was only infecting cats, now it's infecting cats and humans, and that's kind of when it becomes relevant to us. And again, like we said, one of the reasons viruses are so bad is because they are so simple. They are constantly mutating, and it's really hard for us to get ahead of them, which is why every year we're coming out with a new flu shot, and we're coming out with different boosters for COVID. It's because those viruses mutate so quickly that our body does not recognize the old versions of them. All right, so lots of factors that go into these mutations. We've got physical factors, different microbes, ecological factors, social